DM Science and Plants World presents about the Auxin, a plant growth regulator or a chemical messenger. We already discussed about the plant growth regulator in our overview. Do check the description box to find the link for that video. First, we will discuss about the discovery of Auxin, the person who was involved in the discovery of Auxin and isolation of Auxin. During 1880, the person Charles Darwin found the unilateral growth of canary grass coleoptile. Coleoptile is a sheath-like structure which protect the young shoot tip in the grass and cereal. This coleoptile growth in response to light is unilateral. From that, Charles Darwin find the activity of auxin. The term auxin is derived from the Greek word auxin, which means to grow. And this word is first used by the person F.W. Went in 1926. F.W. Went find the activity of auxin in oats coleoptile. Again, F.W. Went also found the activity of auxin in coleoptile, and F.W. Went isolated the auxin. Isolation means separation of auxin from the particular part. F.W. Went in 1928 collected auxin from the agar jelly, and the persons like Kogel, Huggen Smith in 1931 isolated the auxin from human urine also, and they named that chemical as auxin A. Later in 1934, similar active substance is isolated from the corn grain oil, which is named as auxin B. Kogel et al. And that means Kogel and many persons in 1934 found the heteroauxin in the plant, which is chemically called as indole acetic acid, that is IAA. So this is about the discovery of auxin and the persons who involved in the discovery of auxin. Types of auxin. The auxin are two types, that is natural auxin and synthetic auxin. The auxin occurred in plant in a natural way is said to be natural auxin. There are four types of natural auxin. They are the indole acetic acid, that means IAA, indole propionic acid, that means IPA, indole butyric acid, that is IBA, phenyl acetic acid, that is PAA. Second category is the synthetic auxin. The synthetic auxins are the chemical compounds which are synthesized artificially, which has the properties of auxin. There are three types of synthetic auxin. They are the 2,4-dichlorophenoxy acetic acid, which is commonly known as 2,4-D. Second category is the 2,4,5-trichlorophenoxy acetic acid, that is generally known as 2,4,5-T. Third one is the naphthalene acetic acid, that is NAA. So this is about the two types of auxin. Occurrence. We discussed about the discovery and types of auxin. Okay, we'll discuss that natural auxin is present in the plant. Where it is present, that means where it occurs. Yes, occurrence. The auxin is generally produced by the growing tips of stems and roots. From the tip region, it is migrated to the all regions of the plant and it performs the physiological process. Antioxin. Antioxin are the substance or a compound which inhibit the effect of auxin. See, consider the activity, that is physiological action of auxin is cell elongation, means if any compound which inhibit the cell elongation process which is stimulated by the auxin, and that substance or a compound is said to be antioxin. Example for antioxin is 2,4,5-triiodine benzoic acid, that is generally known as TIBA. And the second example is naphthalene. Free auxin and bound auxin. These are the two types of auxins which found in the tissue. Free auxin has the capacity to move out of tissue easily, which means they diffuse easily. Example for free auxin is IAA. Bound auxin are the auxin which are bound inside the cell and they don't have the property to diffuse easily. That means they are not diffusible. Example for bound auxin is IAA aspartic acid. Precursor. Any substance or any compound which are synthesized or prepared should has or have the precursor. From the precursor only any compound or any substance or any molecule will develop. Likewise, here auxin or a type of auxin, that is IAA, is prepared from the precursor amino acid tryptophan in the presence of zinc. Chemical structure. Auxin has the similar structure of IAA. This is a structure of auxin which has the benzene ring, NH group, OH group and oxygen group. Transport in plants. How the auxin, which is, we already discussed that auxin is produced from the tips of root stems and leaves. From that, how the auxin is transported to all over the plant body. Means auxin involved in polar transport. That means polar means it transport cell to cell manner based on the polarity, and it localized the plasma membrane. By that it moved from cell to cell. It also includes the transport in the way of basipetal and acropetal transport. Basipetal transport means it transport through phloem, which is a food conducting tissue. We already discussed about the phloem tissue also and xylem tissue also. Do check the description box to find the link for those videos also. And the auxin transport through the phloem from shoot system to the root system. This is basipetal transport. In acropetal transport, auxin utilize the xylem, which is a water conducting tissue, and through that tissue it transport from root system to the shoot system. So in this way the auxin get transported all over the plant body. Next one is the physiological effect of auxin. Auxin promote cell elongation in stem and coleoptile. As I said before, coleoptile is a sheath which give protection to the young shoot tip that present in the grass and cereal plants. So cell elongation means elongation of internode. Node is the point where the branching or leaf produced. So in between two nodes a portion is said to be internode. Cell elongation involves in internodal region. At higher concentration of auxin, the auxin used to inhibit the elongation of root. Instead, it induced the production of more lateral roots which are endogenous in origin, which are produced from the pericycle. Already we discussed about the characteristic features of root. In that we discussed that roots, that is lateral roots, are endogenous origin, which means they are produced from the pericycle. So at higher concentration, the lateral roots which are originated from the pericycle is produced in higher rate. At higher concentration, it also promotes the growth of root, that is lateral root. In lower concentration, that is at extreme lower concentration only, it promotes the growth of root. The suppression of growth in lateral bud by the apical bud due to the production of auxin by the apical bud is said to be apical dominance. Apical dominance is the one of the characteristic feature of auxin, which means auxin which is produced by the apical bud that result in growth of apical bud alone. Instead, the lateral bud won't grow because the auxin do their physiological activity and develop the apical bud alone. It forgets to take care of the lateral bud. So this is called apical dominance. Auxin prevent abscission. Abscission is a process of natural detachment of parts of the plants like dead leaves, ripen fruits. So, auxin prevents the detachment of plant pods. The other physiological effects are the it responds for the initiation and promotion of cell division in the cambium cells. We already discussed that cambium are the cells which are present in the vascular bundle which is responsible for the secondary growth of the plant. So it check and initiate the cell division which happens in the cambium cells. This property of induction of cell division helps in tissue culture techniques which helps to form the callus. Callus is the mass of undifferentiated cell. Auxin stimulate respiration. It also induces the vascular differentiation. Vascular differentiation means differentiation of cell which are present in the vascular system, that is xylem, phloem, cambium, metaxylem, protoxylem, protofloem, metafloem. Like that, the vascular tissue get differentiated with the help of auxin. What is the agriculture role of auxin? Auxin helps to eradicate the weeds. Weeds are the plants which grow unnecessarily in the cultivation land. If we cultivating one particular edible species, some other grasses which we didn't focus to cultivate is said to be weeds, are the plants which disturb the growth of focused or cultivated plant is said to be weeds. To eradicate those weeds, example Parthenia is a weed plant. So to eradicate those type of weeds, the auxin 24D 245T, that is synthetic auxin, plays a well-rolled eradication role. The synthetic auxin used to form the seedless fruits, that is parthenocarpic fruits like banana, orange and all. That means the fruit found without seeds. It also used to break the dormancy period in the seeds. It also induced flowering in pineapple plant with the help of NAA24D. It also helps to increase the number of female flowers and fruits in cucurbits. That means Cucurbitaceae family member plants. So this is about the plant growth regulator auxin. Thank you so much for watching.